Okay, so what you see on the slide right now is a color-enhanced electron scanning microscope image of a tardigrade, or a water bear. And what's more astonishing, aside from its looks, is the fact that it was provided by NASA. So why would NASA give us this image? Tardigrades, like Dr. Glazer was saying, are one of the most capable creatures. These are the only organisms on planet Earth known to withstand a vacuum in space, and we use them in our study. So good morning, my name is Srikanth, and I'll talk to you about the necessity for metabolism and memory maintenance in tardigrades. So I began this project June of last year at the Krasnow Institute over at George Mason, specifically the PB&J lab. And then I continued that at Thomas Jefferson in the fall and spring semesters. So the purpose of our experiment. There's a lot of work being done on neuroscience right now. And even though there's a lot of work being done, it might be to our advantage to ask more fundamental questions, to look at the fundamental aspects of maybe memory. So we introduced a novel approach using cryptobiotics and neuroscience by merging the two fields to answer this question. Is memory static or dynamic? So is it structural, or does it require some sort of active processes? And I'll talk to you about that in a little bit. So, overview. I'll go over the background, experimental design, methods, some of the results we got, analysis, and conclusion, and you can follow along at the bottom. So, what is a tardigrade? So, tardigrades look like that. That's another scanning electron microscope image. They are less than ½ millimeter long, they're microscopic, and they're generally transparent. You can find them in freshwater sediments, mosses, anything that's kind of aqueous. But the main reason we chose tardigrades is because they can undergo a state of cryptobiosis. And what is cryptobiosis? So we call this tunning, short name for it. And essentially what they do is when environmental conditions become too extreme, they will enter a state of cryptobiosis, which means they suspend their animation, and they can stay in that indefinitely until conditions become a little more favorable. So for example, if it's too dry, they might enter the state, or if it's too salty, they might enter the state. And specifically in our experiment, we dried them out to get them to get in the state and then tested their memory like that. So memory. There's 3 stages of memory: encoding, storage, and retrieval, and we primarily focused on storage. Maintenance of memory is one of the things that us researchers don't really understand. There's 2 kinds of memory, short-term and long-term, and we focused on long-term memory. And this is for 2 reasons. 1, long-term memory, short-term memory cannot sustain generally the time it takes for these animals to enter a tuned state. And 2, long-term memory is associated with LTP, or long-term potentiation, which is essentially structural changes in the synaptic level of the neurons, which is what we wanted to see. So this is what we hypothesized. The maintenance of long-term memories does not require ongoing neural dynamics. In other words, this memory will sustain this tuned state because it's purely structural. So think about it like this. If you had a computer and you write a memory, you write some sort of program, and you turn the computer off, will that memory still be, will that program still run after you turn it back on? That's the kind of question we were asking. So for example, if it was a solid state flash storage, it would. If it was RAM access memory and RAM processing, it wouldn't. And that tells us the structural dynamic. So because this experiment was very vast, there are a lot of little things to do, so we subdivided into multiple separate investigations over the course of the past few months. And it's still ongoing right now. The primary thing I focused on was tunning, and this was my semester at TJ in the fall. And generally, the species that we use is Hypsibius digerandi, and they're not the most capable of all the tardigrades. They're not the most capable species. They generally have a 40 to 50% success rate. With the protocol I developed, we get a phenomenal 80 to 90% success rate, and here's how we did it. We take the petri dish, this is general lab protocol, make the petri dishes, set the glass bell-shaped desiccation chamber, and inside that you see a beaker, and that beaker is filled with saturated potassium chloride. And this is so we can regulate the humidity at around 87 to 93%. So then we take the tardigrades from their home solution, put them on the petri dish in a droplet, and then place them in the desiccation chamber. So what happens is, over the course of 48 hours, this droplet will evaporate, and the tardigrades will be in the agar without a droplet, but still maintained at a 90% humidity. Then we take them out at this stage and put them in regular lab environment, which is about 40%, give or take 5% relative humidity. And by this time, they are completely tonned, and to bring them back to life, all we have to do is add water and wait 5 to 10 minutes, which is really astonishing. We're also working on conditioning. It's currently taking place at Krasnow. What we do is place the tardigrades in a droplet in the center, place food with the tardigrades, and then have some sort of volatile chemical on either side, on one of the sides. And we start them, we place them, and then we run some chemotaxis assays to see do they actually go to the acetone afterwards. And we're supervising protocols for this, and hopefully that'll be done soon. So a lot of preliminary testing had to go into creating protocols for what we have in ton of conditioning. So I'll talk to you about what we did over the summer. So chemical bias assays. We had to make sure that these tardigrades did not have any innate bias towards any volatile chemical. So what we did was we took that template, we put acetone on one side and ethanol on another, and we saw where they went. We created an algorithm to quantitatively label what we were seeing, and this is what we saw. So after about 30 trials, these are the attraction indices, and if you run a regression on it, you get an average of 0.017, and the error bars far surpass what we see, which is statistically insignificant, which is exactly what we were looking for. So to make sure that, so how do we get the concentration? How do we know what concentration to use for this test? We ran repulsion assays, and we ran repulsion assays with acetone and ethanol. All we did was create various concentrations of these chemicals, place the tardigrades next to them, and see if they go back, go away, or towards the chemical. And this is what we saw. So the top is for acetone and the bottom is for ethanol. In both cases, we see between 5 to 10%. There's some 0 point, which is the concentration that we focused on. Anything higher than 10%, we see some nonlinear regression, or some nonlinear trend, which we didn't want at all. That would screw results. Specifically for the ethanol, we thought that they might be getting intoxicated through diffusion through their cuticle, so we really wanted to stray away from going to high concentrations. We also did some starvation assays, and starvation assays helped us create a protocol for conditioning to make sure that they were actually starving, they were learning to associate a chemical with it. So we use a software called Metamorph, and what Metamorph does is it takes pictures of the tardigrades, and because these are transparent, we look at the opacity of their gut, and then we compare that by creating a ratio between a starved tardigrade and a regular tardigrade. And this is what we saw. After 18 hours, there was a significant difference with a p-value of less than 0.05. So after 18 hours, we know that the tardigrades need food, they want food. And that's how we know that they're starved. Just on a side, we also did some neuronal labeling to get a better understanding of the anatomy, the neuroanatomy of the tardigrade. So here's some of the pictures that we saw. So on the top, you see DAPI labeling. And this tardigrade is facing this way, and that's the head. And what we see is 4 very main ganglia, and near the head we see a cluster of neurons around that, which is somewhat analogous to what we see in vertebrates too. Bottom left, we see immunohistochemical labeling. This was just to label some of the parts. The eye spots are labeled in red right here. And finally, we did some anti-HRP, and that's essentially showing us different kinds of all the neurons in the tardigrade head. So this is an ongoing experiment. We're still finalizing the protocols, and we're looking at conditioning a little more. And once we finish that, we're going to continue to do the train, ton, test for memory, and we'll see how those results turn out. Some of the recommendations as we move further, we're trying different chemicals aside from acetone and ethanol, stuff like vanilla extract, semen extract. We also want to try different species or different kinds of memories. What makes this project really interesting and applicable is, well, the applications. What can we do with what we learn? And there's really 2 or 3. So the first one, understanding cryptobiosis a little more helps us with cryopreservation. Tissues, information storage in tissues is starting to become a more prevalent thing in the world. 2, we want to use the tardigrade, we want to put the tardigrade out there in the research world as a model organism because of its unique characteristics, because it can undergo this ton state. And some of the applications in medicine. 3. Understanding the biochemical basis of memory allows us to target drugs, prevention and treatment drugs, to specifically target either the structural aspects of memory, parts of the brain, or two, maybe target the bodily processes that affect what the brain is doing. So I'd like to take a minute to thank some of the people who were involved in this project. Dr. Dumas of the PBNJ lab, if you want to raise your hand. Mr. Hammond, who guided me here at the TJ lab. I don't know if Mr. Hammond's here. And Ashley Coker, who's not here with me, with us today, but she helped train me over the summer. And Mr. Pierce and Mrs. Luxembourg for taking care of the whole mentorship program and allowing me to embark on this project. And of course, family and friends for their patience and support. I'd like to thank the PBNJ lab over at the Krasnow Institute and the Neuroscience Research Lab here at TJ for letting me have this space. Literature side. Okay, do we have any questions? Okay. Yes.
Was there actually a long-term and short-term memory in a target? How do you know where the food is long-term or short-term?
Okay, so long-term memory is essentially memory that lasts longer than like maybe a few minutes or hours. Because what we were doing with the tardigrade was hunting them over a course of 48 hours, we put the tardigrades in that chamber for the weekend, so the droplet can slowly evaporate and they can get accustomed to the environment. Short-term memory does not last that long, so we know that they were learning long-term memory, and if that's sustained, it was only going to be long-term, not short-term memory. Yes.
Is it possible that the cutting stage is still providing some kind of sustaining preservation of
Yes. So at the ton state, generally the consensus is that metabolism ceases, everything pauses, but measurements would show maybe a very, very low, 0.0001% or so. That's what the literature states, but because it's so low and insignificant, we're assuming that there's really nothing major going on to sustain what's going on in the brain, or in the ganglia. Yes?
Do you have a sense of memory capacity these days?
They're obviously very small critters. They're very rudimentary, so we're assuming that it's only going to be very primitive memory, nothing elaborate. All we're doing is testing their associative memory characteristics. We're not testing any like logical thinking or executive processing, so it's only associative memory. Okay, thank you.
Thank you, Srikanth, for the excellent presentation. Now I'd like to introduce Parth Chopra, who will be presenting an agent-based model on the outbreak, spread, and containment of tuberculosis.